Well, I'm just asking you, what, in the next whatever time you have, that it's great to see people on the call. What's your goal for the panel discussion? I mean, there are lots of comments about lots of things, like the future of the PDB, et cetera.
Well,
et cetera.
I was, since we've started down the road of talking about the importance of model validation broadly, and I know you have opinions on this matter, I thought you might want to jump in now.
Well, not on the particular, yeah, thanks for, but not on the particular one that Andrzej described. I mean, I think that what he described is a very difficult enterprise to bring together all these different kinds of models, which is admirable in its collection of expertise. I think there's one point that maybe he said that, Andrew, you said that, but it wasn't totally clear to me. To what extent does one have to focus efforts like that on a particular goal or a small set of goals, so that the models can be filtered with respect to whether or not they're actually going to be useful, quote unquote, to a particular goal? Yes, uncertainty is important. Yes, data quality is important, all of that. But what I'm missing a bit is the goal orientation as to what are we doing with these models, because models are only useful and only if, well, okay, let me stop saying. I mean, there's a question of whether the models are correct is number one. The other one is whether they're useful. And I don't mean the banal statement that all are incorrect, but some are useful. What I really mean is that in science, we make models of the world and you want to be able to apply them. The pancreatic cell example is a nice example of where you have a medical focus. And I think that's one point I really got. Let's bring these together to solve a problem. But in general, I'm asking, and this also affects the future of the PDB, what's the extrapolation from all the data collection and et cetera, to what we're going to be able to do in the world? And there's some obvious things for proteins like engineering and embedding proteins and protein machinery in other technical devices and so on that will come in the future. Anyhow, I'm saying too many things in one go.
So I think one thing that I've been thinking about is
oh, that's Jane. Hi, Jane.
Hi.
Jane, your hand is still up, but is it really still up?
Sorry. Sorry. No.
Okay. So in a way, that I think it's really interesting that we've come to this 50-year anniversary of the PDB at the same time as the AlphaFold announcement of their accomplishments. And we have, I mean, there's a temptation perhaps to say the PDB should do more and everything and this and that, but supposing we say, what are the most important things that the PDB should do, and the data, what kind of annotations need to be done to meet all the new challenges that we now know about? Maybe that's a way to frame this. So at what level of data quality, how do we actually describe the data quality? How do we make it so that one of the reasons that the models originally were taken out of the PDB is people were using models as if they were experimental things, and then we would never have the accomplishment of AlphaFold because it would all be mixed up. So the question is, what to me would be, how should the data be described and annotated so people know what they're getting? Is there more that the PDB should be doing just with what it's doing now? I have that question that I'd like people to explore. No takers?
Wayne, do you have thoughts? You've obviously reflected greatly on the first 50 years. What about the next 50 years?
Well, I'm not very far-sighted, so I want you to keep up what you're doing. But and
I fully intend to do that. The question is,
I think,
what do we need to do?
And I'm not up to speed with Charlie's approaches with all these vastly different kinds of information being coupled together. How you take good use of I don't know, these ordinary differential equations relating one thing to another and how that relates to structures and so forth is a little beyond me, but I think the direction of trying to get more global is really great. I think it was Chris who said embedding these things into their milieu, so to speak, is really a right approach. And try to model the cell, a goal that many people have had for a long time, becomes approachable in the next 50 years. So I think we have to do it.
But should we think in terms of, where does the PDB and other databases take over and make it so that we don't get into a thing, and this is just my prejudice, okay, I will admit to that. We don't want to ruin what we already have, but we want to encourage things to happen. And we need to figure out how to do that. I think Ava has a question, a comment.
Actually, Jane was before me. I saw her hand before me.
Jane,
So
did
I'll
you?
go on.
Okay, mine is fairly simple. I was just saying that one of the things that I think the current system could do better is actually also going in the other direction. That when we talk about uncertainties, we should be doing it locally. And you do that, but the information is very buried and hard to get at.
What do you mean by that, Jane? I'm
Well,
not sure I understand.
it's really obvious in cryo-EM, but it's true in crystallography as well. The resolution isn't actually a very good measure of the local uncertainty. And
You're absolutely right.
It's very clear in cryo-EM. In crystallography, at least at reasonably high resolution, it tends to be extremely local. That you have one side chain or one little loop that you really couldn't see. And making that more obvious to people. We have some of the data for that, although most uncertainty methods just deal with normal mode, normal distribution type uncertainties, which are not the major effect in these structures. The major effect is when you have another rotamer or a different conformation of the backbone. And just making that more obvious. We've been trying to do that all along in our validation, and we now finally have a way of showing it in the data set, several gigabyte data set for a small part of the protein data bank, about 10%, I guess, that actually is up on Zenodo where people can use it. And we're very, we want to publicize that and have people use it and see what they can do with it. But certainly one could, in principle, do it for the whole data bank. But, how to make this evident, how to make it choose thresholds and so on is difficult. So it doesn't have to be done the way I'm saying, but I think we need this local uncertainty somehow.
So I think that you're absolutely right, Jane, that there are challenges with the current system that the Worldwide Protein Data Bank validation software is using. And some of them have a statistical basis in that there were implicit assumptions regarding normal distributions, et cetera, which we know to be false. And there is no easily accessible measure of local uncertainty, and we are aware of this, and we are working to improve matters. So please watch the space, and of course, please contribute as opportunities develop for making contributions, and I make that request not simply to Jane Richardson and Dave Richardson, but to anybody in the community who feels that they can contribute.
Great, thank you.
Thank you.
I think Ava had a question.
Yeah, so I just wanted to ask, what is the thought? So we've been mentioning, of course, AlphaFold, Rosetta Fold. I don't know about the rest of you, but I get asked all the time, how is this going to affect your work? And I say, you know, for me, working on very large things where it's very hard to get to better than 4 angstrom resolution, having prediction of the different components of my complex that I can trust, at least as a starting point, is magnificent. But in any case, Rosetta Fold for sure. I don't know what AlphaFold will be doing. These are freely available. Anybody can go put in a sequence. You know, I think high schoolers are going to be told to do this for projects and things like that. There may be a real flood of structures coming out of it. How many of these are going to be deposited? How is the PDB is gonna deal with that tsunami that is going to come? So that's one question that I had. I don't have the answer, of course. And the other is concerning the modeling of the cell. I think many people is aware that the next revolution on the EM side of things is electron tomography. I think the field is not really there yet, but it will be. Before I retire, there's gonna be structures of large sections of cells with enough detail, and I think signal to noise, to be able to start docking in structures that are available and placing everything in context. And again, do we need, what kind of database will be the one that will contain that mixed kind of data, where there's segmentation, gray levels, models docked in, and things like that. So those, sorry, no answers, just questions on my side.
Oh, thank you, Eva. So let me respond to part one, and then let me defer to Andre for part two. As Helen explained, the current posture of the PDB, and has been for many years now, is that PDB structures require an experimental study of a biological macromolecule in order to be included, of the biological macromolecule in question to be included in the PDB. That's unlikely to change in the immediate term, but it is clear that we need to make it possible for computed structure models coming out of Rosetta Fold, coming out of AlphaFold, and being stored in other places, and I'll come back to that in a moment. They need to be able to interoperate with structures from the PDB. Now, fortunately, quite a few years ago, the Model Archive, which is run by Thorsten Schwed at the Swiss Institute for Bioinformatics, adopted a data standard for computed structure models known as Model CIF, which is based on the PDBX MMCIF data standard, which is used for all PDB structures. So anything that has been, any computed structure model that is produced in the Model CIF format can, in principle, interoperate with structures from the PDB. Fortunately, all of the AlphaFold DeepMind computed structure models are in Model CIF, and there is a setting in Rosetta Fold which allows for generation of Model CIF output. So, there's no reason in the immediate term why these models can't, these computed structure models and the models derived from experimental data cannot actually be used together with various software systems. Andre, would you like to jump in?
Yeah, I have some comments here. So first, I'd like to start by noting that structural biology is uniquely positioned among various subsets of cell biology to be the nucleus towards building a really useful cell map. Why? Because it's the structural biology models that are closest to reality of all models. There are atoms, and they have positions, and their positions change in time. That's a more concrete and closer to reality type of a representation than, say, molecular networks or ordinary differential equations. Of course, these other representations have their role as well, but because the structural biology types of models are closest to reality, I think it's the structural biology domain and the protein data bank representing the output of this, of our community, that is uniquely positioned to nucleate the cell biology. And that's starting to happen. And on the technical side, Helen, again, had this idea of a federated archive of various types of data, different types of data in different federated resources that are linked together, but they're linked not just haphazardly, they're linked through structural models that result from these data in the process of integrative structure modeling most often. And so one can imagine that being generalized and strengthened and made even broader than it is currently, including mass spec types of data, and various kinds of imaging data, spectroscopic data, and so on, some of which are already part of the integrated methods federation under the WWPDB. So I think, the question is, in my mind, is not whether or not that's a good approach. I think it is. The question is exactly, at the detail level, how this should proceed. That's one comment on Helen's original question about what should the scope of the future effort of the PDB be. I have other comments. I don't want to monopolize the discussion.
So Helen, would you say more about the data, about the opportunities for federating data? So the Worldwide Protein Data Bank is already a data federation in the sense that it includes the PDB, the biomag, ResBank, and the electron microscopy data bank. But we're thinking on a much larger scale in terms of being able to integrate and interoperate with data coming from different sources.
So we had a workshop, I can't remember, it was about 3 years ago as part of a Biophysical Society meeting on integrative modeling and what to do and how to think about it. And as Andre showed, there's many different experimental methods that go into creating the restraints that are necessary for doing integrated models. So the thing that we came up with at that meeting was the idea of, for each of these methods would have its own database, okay, where they would be free to collect whatever information they want, however they want to do it. And the only requirement would be that the restraint data would be able to be passed to the, in the case of integrated models, you want to have all the restraint data coming into the structural databases. At the time of that meeting, we said, again, things are evolving very rapidly. We said there's the PDB, there's the model archive, and then there's this integrative database that had yet to be formed, and it is now formed. And they would be in the center of the circle. And all they would have is coordinates, appropriate metadata, and the restraints, okay? And then on the outside of the federation would be all these different experimental methods, and their only commitment would be to have standards, and that the standards would either be similar to MMSIP or interoperate with MMSIP, however. So at the time we did that, I had no idea what was actually going to happen. We already, as Stephen said, we already know about BMRB, which is for NMR, and EMDB, which is for electron microscopy. But since that time, there have been 3 other efforts. And actually, the small angle scattering has something. And now FRET has created standards. Crosslink HDX has standards. And I know from various reasons that EPR is setting standards using what we said as the basis for doing this. So we already have several methods now that are forming standards, like the idea of nucleating their community, working and interoperating in some way with the PDB, which is now happening with the ones that had been formed earlier. So that's the idea, is that the PDB wouldn't contain all the spectra and all the raw data and all that. That's just too much. But if all these communities did that, and they provided the restraints in a way that was standardized, that we all agreed on, we could have a greatly expanded set of structural databases, whether it is the PDB and Model Archive. Right now, that's the way we recommend it. That could change because everything changes, okay? And we have to be prepared for that change. But I think we have a framework now that will work for our community and will also provide a model for other communities. There may be a situation, and I think there is a situation, where other communities can step up to the plate and make their own kinds of databases that would then interoperate with this particular federated group that we came up with. But that's where we are. That's what we mean by federation. So the PDB would not contain all the data in the world because we couldn't do it, right? I mean, nobody could do that. But there would be standards that would allow all the data to move around. And that's actually happening, to our great surprise.
But the what's changed, if I may jump in and be provocative, what's changed is that AlphaFold has just released models for about 360,000 protein sequences. So that's double the current size of the PDB in terms of the number of entries, number of predicted structures, versus the 880,000 experimental structures in the PDB. And by the end of this year, they're going to put computed structural models for all of UniProt 90, which is 130 million sequences. So that's a huge jump in terms of the just the size of the amount of data that's going to be available. And then we have and I think this was one of the points that Chris was driving at earlier, and I'd be grateful if he'd jump in. Then we have the issue of where are all the models of cells or models of organoids going to be stored, and what are the standards going to be used to capture that information, particularly because these models are going to be continually revised. There are going to be improvements in the technology, and a new round of modeling will be, a new round of calculations will be reported. So there will even be versioning of these models as there is actually a versioning of the PDB. It is now possible for the depositor of record who, with the same experimental data, can choose to produce a superior set of atomic coordinates and put those into the PDB, and that becomes a new major version of a PDB structure. And we introduced that so that people could go in and clean up errors from the past, and also take advantage of modern techniques. And I don't have an answer for how we're going to house models of the complexity that Andre was describing for the pancreatic beta cell, but as Chris said, there's no doubt as to the medical importance of type 2 diabetes. Huge numbers of people around the world are affected with that as access to calories grows.
So I'd like to comment on this. I mean, the PDB doesn't have to, I mean, maybe Andre doesn't agree, but that's okay. The PDB doesn't have to house cell models, but the PDB can provide the framework for how to do that, okay? Because we know now how to do that stuff. So I think we can, the structural biology community can lead in this. It's not an impossible problem to solve. I have my own prejudices about what should or should not be in the PDB, but if we have a framework for how to do it, then it's very straightforward. Certainly for the issue of the models produced by AlphaFold or any of the other programs and systems that are evolving, I think that's very straightforward. We have that framework all in place. The only part that I think is difficult, really difficult, is we have storage issues, as you pointed out. I mean, they're resource issues that have to be solved. But at least there's a way to do it. And in the case of the modeling, cell modeling, I'm not sure that PDB should be doing storing cell models, but it can provide the framework for how to store cell models. And I think we're very far advanced in that way of thinking. So I think the PDB should be, the PDB is leading and should continue to lead, maybe not by doing all the work. I don't think that makes sense, but by providing the knowledge for other people to do the work.
Yeah, so maybe Wayne, you go first.
My comment is very simple. I think that as to the hundreds of millions of AlphaFold structures on the horizon, I think you at the PDB should keep your standard that you've had, that you're not going to be a repository for models. And I don't believe that there's anything like enough validation of AlphaFold to say it supplants the experimental definition that you've had for some time. So I think you can certainly punt for a while, but there is a need to make sure that the modeling doesn't get sort of siloed, so that there's a set of models here and another set of models there, and so on, that everybody has to consult all of them. I don't know how that gets resolved, but it's another big problem, and it'd be great if PDB could take a leadership role.
Could I comment?
Yeah.
Yeah, and I just want to comment on you can go first, Helen, if you want.
All I was going to say is it doesn't have to be in the PDB, but because there is already a model archive that completely interoperates with the PDB. It could be very straightforward to put these, in terms of intellectually straightforward. We have to talk about resources and all the rest of it, but it is a straightforward problem that has been solved in terms of how to do it. The practicalities is something else.
So I'd like to make two comments. One of them was a 1 year, then there was a 10-year comment. The 1 year comment has to do with the models in the AlphaFold, the Rosetta fold, and so on. And so it's not much of a debate because needed. There's two obvious points. First of all, this is just a page from Uniprot, and so the work behind the scenes to put these models up in at the EBI in an archive. I just dialed up two of them. Here's one of them, TF2D, and there's a model, and you know, some models are good and some models are crap. This one is on the crap side, and you can see that. I mean, the good news about AlphaFold is that they actually have a confidence index. I still have to understand exactly how they define it, but you know, suspending disbelief for the moment that this pair of helices and this thing here are of higher accuracy than the rest of it. Or overall, the model is just maybe useful. You know, it's of very limited usefulness. I just call it crap. Okay, so, but I don't know what the spectrum is. We're going to find out. So that's the first point. And the footnote to that is that they're already doing it. They have it up there. Whether PDB does it or Uniprot does it, doesn't matter. The models are there. And Rosetta Fold will also have models out there, and there may be others. I don't know. So and of course, having them linked to sequence and structured databases is a good idea. So call that done. The only thing that the community has to understand, you know, how to use those models and which ones not to use for which purposes and which ones are extremely useful. Like, for example, the example I showed where you have a predicted model if it's good, and you could do molecular replacement. And there are lots of other examples like that. What's overhyped right now is doing drug design on top of those models. Okay, so those are my comments about the model usage. I don't think between PDB and Uniprot we have to agonize over it, consider it done, because they're available. You may want to raise a flag in terms of the quality and so on. Okay, so that's number one. Number two is my 10-year comment is that, and you know, I mean, you're going to be taking up some of these things on your PDB advisory board and so on. There's lots of details, as just discussed, that you have to, and all the comments that you made, Helen in particular, and Stephen, is that you have to take into account exactly what are the standards, the data standards, where do you draw a line, how do you flag things and so on. But allow me to just make a comment about, you know, not 50 years, but 10 years. I mean, the information landscape, of course, is rapidly changing out there in the world, and also in science, things getting incredibly more linked together. And of course, Andre has a vision of how to do that in terms of models that are available that I think is on track overall. But if you think of, you know, how information is going to be organized, I call it a mental walk. Humans want to take a mental walk in the world, including in science, and the mental walk that they want to take in analogy to the human brain is question-oriented and also context dependent. So take, for example, some of the recent developments in Google searches. You search something, and what you get is just you know, a list of things. Well, they're trying to go beyond that, right? And now you get a list of questions. Often I found this extremely useful, not to advertise Google, but you know, they have inferred questions you might have in your mind that are relevant, and they actually give you the questions and give you some answer. It saves you time in many cases, actually, right? So now imagine scientific questions. So one potential vision is two parts. Okay, I'll try to keep it brief. One of them is that whatever you do in PDB or Uniprot and so on, try to serve up the information with questions in mind. Phrase the question or anticipate the question, and then serve up the information in a way that addresses those questions. It's more than just having the data, and it's more than just showing crystallographers and a few drug design people what's there. It goes way beyond that. But you'd have to be aware of what those questions are, number one. And number two is the context. And the context is difficult, but you just talked about that. But I think that there's going to be, in the future, 10 years, 15 years, 20 years, or maybe 5 years, much, much closer integration of everything. And I think there's going to be technical developments in the software world that are going to integrate things much more closely. Think of the genome databases that have annotation tracks. That was a good idea, and it works very well. And they can add more information to it. Are there annotation tracks in the PDB? Well, maybe it's not that easy. So I'm just trying to think of how can we think through the next 5, 10, 15 years in terms of the really breakthrough development that are going to happen out there and also in science, linking information that's question-driven and context-driven, and then try to move in that direction in incremental steps.
Thank you, Chris. I notice when I shop online that if I click on an item or if I buy an item, I'm frequently told customers who liked the product that you've just picked out also bought X. And so it's been my plan for a while on rcsb.org when people do searches and make selections to say, you should be aware that in addition to this structure that you've just picked out, there's a whole bunch of other structures in the PDB that are very closely related, either in amino acid sequence or because they're known interactors. Those are the sorts of things that we've been planning to implement. And with the advent of so many computed structure models, there's an opportunity to do this much better, because we're no longer going to be limited by what happened to have been worked on by experimentalists that ended up in the PDB.
If I may.
Andre had a question.
Yeah.
Actually, it was more like a comment on what Chris was saying, and then on model assessment. Actually, I think, Chris, we are kind of almost saying the same thing, just in different words. Different types of models exist. Each one exists for two reasons. One is it's possible to get it. There's data and methods to compute it. But the other reason, perhaps more important, is that it's useful for something. And different models are useful for different questions, just like you said. And therefore, a really successful and general effort in presenting all conceivable types of models about some system of interest, such as the cell or the bodies, would address what you were saying if it was done well. It's just basically the same thing. On model assessment, I would say real quick, because I do hope there will be an actual effort, again, out of PDB or RCSB or WWPDB, I don't know, on unifying model assessment across the different types of models. The mental picture that's required for this is actually quite simple. Its importance relies on the fact that once you know, if you know accurately the uncertainty of a model, it doesn't matter how that model was computed or determined. You just have to consider that uncertainty. It doesn't matter how you got it, right? So the uncertainty can be defined most generally, and therefore, if you want to, you can apply it to each of the existing and any method for model calculation that I can imagine, can be described most generally by some measure of variability of some aspect of a model you care about across all models that are sufficiently consistent with the input information. Right, let's a simple example, NMR structure ensemble has some variability in it, and that's potentially a good measure of uncertainty, reflecting both the imperfect input information as well as heterogeneity in the sample, problems with computational method, model representation, everything, right? But that's a measure, and that sort of thing can then be applied to all kinds of methods, if you wish, and could be done in a Bayesian fashion in principle. And if that's actually done in practice, then you will have had that beautiful general, across the board type of uncertainty estimate that everyone should learn in, I don't know, high school, certainly in college and graduate school, how to use any scientist for whatever biology they do. And PDB can be really instrumental in doing this for structural biology and hopefully beyond to some degree.
I'd like to warn just about a silo thinking for PDB. I mean, I had one experience, this is just an example, in NHGIR about pathway databases. There were all these different pathway databases, and we did one that integrates all of them and so on. That the NHGIR, as a funding institution, was saying, well, wait a minute, let's make the whole thing more efficient. Let's have things more assembled and more efficient acquisition of knowledge that is then made computable and data ready. So I think that, thinking about the future of PDB, which is, I guess, part of the topic here, it's not clear to me that the PDB as an independent organization will necessarily exist. It may, but I think it may well be that there's a consortium of scientific databases as well as knowledge bases and computational tools that use them and develop on top of them. And you may want to think strategically, beyond any particular person being involved and so on, even though you've got pioneers in the room here, of where would you go in terms of an integrated landscape of information that isn't necessarily just PDB continued, but some new version of it that's linked in some way. Maybe it is PDB, but I'm just saying, let's be open to a landscape that may be radically changing and be a leader in that.
Agreed. Jane has I think may have the last word.
Oh, this shouldn't be the last word, but it's a caveat on Andre's elegant description of putting together all the uncertainties. We thought we had some of that back in previous decades in doing validation or assessment or uncertainty. And it all fell apart when people started refining the criteria that we were measuring. And so if you get a perfect Ramachandran score at low resolution, it usually means that you refined them all away, not necessarily fixing the problems underneath them. It's a real big problem at the moment.
Yep, that's absolutely right. It was a process of selection. If you punish people for too many Ramachandran violations, because you expose them in the validation report, then people will seek to minimize them at the expense of some other part of the structure, and probably rendering the structure less accurate in terms of reflecting what's really going on. And we've absolutely seen that. Yeah.
That's definitely one part of it. The other part of it is that with this small amount of experimental data, you have to use more restraints. So I'm not trying to get people not to do that. They have to do it.
Yep. And the restraints have to be
not put on restraints that have multiple correct answers, multiple minima, which Ramachandran does. But definitely putting in things like bond lengths and angles and whatever you can. I don't know the true answer to that, but we've found that making good validation is sort of like humans competing with the bacteria. You know, the community's evolution will probably win most of the time, but, but we do have some new ones, and we're trying to do new things. But you can't always just define it all and then sit still. It's got to be.
Yep. No, and we are keenly aware of the need to continue to improve the validation, and have taken a series of steps. Perhaps baby steps to
I imagine you'll get Kablam in soon.
Yeah. Thank you to all of the speakers and those from the audience who participated in the panel discussion. Helen, are you agreeable that we should draw this to a close, being respectful of people's time, and then just do the acknowledgements?
Yes, I think we should do that.
Yep.
Yes.
Okay, so we do need to end bang on 3:30 because there are other things happening at ACA that we need to respect, and we just need to be respectful of people's time. So let me share my screen for the last time, and I will make a few comments and then invite Helen to have the last word. So, boom. I hope everybody can see my slides.
Yes.
Yes, very good. Okay, so I want to begin by thanking all the speakers who were too numerous to list, and then to thank all of the session chairs, Helen Berman, David Rose, Natalie Stranadka, and Ray Zhao, and to particularly thank David and Natalie and Ray for serving as co-organizers with me to put together an absolutely terrific 2-day program with talks that were first rate, great participation in terms of the level of participation and the level of engagement that we saw from the participants. No, just going back to my introductory slides from yesterday, none of this would have been possible without the 50,000 plus data depositors who've put data into the protein data bank, and some have put data into BMRB, and some have put data into EMDB along the way. The many, many millions of people who are consuming PDB data and downloading X-ray structure factors, EM maps, and NMR spectra information. The PDB is in no small measure where it is today because of Helen's vision in the early 2000s, culminating in 2003 with formation of the Worldwide Protein Data Bank Partnership, which initially included the US RCSB Protein Data Bank, that I now lead, Protein Data Bank in Europe, Protein Data Bank Japan, and then more recently accreted the Biomag Res Bank, and just this year the Electron Microscopy Data Bank joined. We are, of course, indebted to funders around the world, both public and private philanthropic, and the individuals, taxpayers, donors, etc., who contributed to that to actually make it possible for these 5 data centers to interoperate with one another and safeguard the world's public structural biology information, and deliver it at no cost and with no limitations on usage. You may be interested to know that we very recently adopted a new license arrangement for PDB data. It's now being distributed under the most permissive Creative Commons license, the CC0, which allows anybody to download the information at no charge and with no limitations on its reuse. And PDB data are being reused by more than 400 external data resources at present, greatly increasing the reach in terms of the millions of data consumers who are able to take advantage of PDB data for research and education. Again, I thank the sponsors of PDB50 who contributed to the Worldwide Protein Databank Foundation to make this series of PDB50 events possible. And I also thank the individuals who contributed directly to the American Crystallographic Association in support of this transaction symposium, and their logos were displayed by David Rose earlier today. Upcoming PDB50 events include a focus on enzymes at the American Chemical Society fall meeting on the 25th of August. On the 6th of October, not listed here, but soon to be publicized, is a Biophysical Society celebration of PDB50. The organization of that event is being led by Guy Monteleone, longtime contributor of NMR structures to the PDB. Protein Data Bank in Europe will be putting on their celebration of 50 years of the PDB virtually. In fact, all these events are virtual out of the EMBL lab in October, spanning 2.5 days, October 20 through 22. And then the RCSB Protein Data Bank and the Protein Data Bank in Europe are collaborating with the Royal Society of Chemistry to put on in November 16 and 18 a training session on effective utilization of PDB data. And finally, our colleagues at Protein Data Bank in Japan will be hosting at the annual meeting of the Biophysical Society of Japan a 50 years of PDB and the frontier of structural biology in Asia on the 24th of November. And I think at that point, we are all going to be completely exhausted by the extra work that we have welcomed in this 50th year of the PDB. I am sorry that because it's also the year of the coronavirus, that we are not able to do these events in person. But the silver lining of doing all of these events virtually is that far more people have been able to attend than would have been able to afford to travel to the meeting. So there is a plus to the new normal that we're in. So, and lastly, I need to thank the staff of the American Crystallographic Association, specifically Kristen and Christina, who've taken such excellent care of the speakers and of these two sessions in ensuring that they went off without a hitch. Helen, over to you.
Okay, so many of you may not know, but we have a foundation called the Protein Data Bank Foundation, WW Protein Data Bank Foundation, whose specific mission is to do outreach and education about the PDB. As you may or may not know, the funding for outreach and education for all of the sites is minimal. And so this foundation is how we are able to have the kinds of meetings that we've been having this year. And so I would urge you to take a look at the site and if you can contribute so that we can continue to run these kinds of meetings and workshops that we've been doing. And thank you everybody for being a part of this whole celebration. And with that, we can end our session.
Thank you and be safe.
Okay.